एक ही अकेला प्रेजेंट है यार। उनके साथ आएंगे। पीएचडी का अवेलेबल नहीं है। Already 10:30. You have to start the other session. हमने प्रपोजल दिया था कि बायोकेमिस्ट्री को इसमें वेटरनरी से मिला। नोबडी हैज टाइम नहीं होता है कि कब कौन सी शाम को ऊपर ऊपर आता है और ऊपर ऊपर मीटिंग हो जाती है। बात नहीं जिनको बुलाना होता है।
We could finish only two presentations. There are, I find in the list there are 6 more. Yeah, 6 more. So, Saini Naveen is here.
Music বন্ধ কর, music বন্ধ কর।
Then Saini Naveen is here. Fodder-free culture of primordial germ cell. Feeder-free culture of primordial germ cell of chicken. Is he here? Next will be comparative study on viability percentage of prenatal antral follicles retrieved from fresh and cryopreserved ovaries. Please come. So speaker, please restrict your presentation within 5 minutes, followed by 1 or 2 questions, so that we can finish our job. Please start.
আমাদের তাহলে সংকল লাগবে বড় করলি তুই আর তুই যখন গেট কর আর ওপর নিচটা ও লাইট দিবি
Myself, Dr. A.M. Kotraj,
বলছি ওই লাইটটা দে না
A very good morning to all. I am Dr. A.M. Kotraj, Professor and Head Veterinary Biochemistry, Veterinary College, Bangalore, Bangalore. I am here to present my students' work, thesis work, that is Takale Devanand, who has worked with Dr. Ramesh and Dr. Nandi sir at ICR NANP. The research work is comparative study on the folliculogenesis parameters of pre-antral follicles retrieved from fresh and cryopreserved ovaries of sheep. Next please. So in 1918, they have published a paper that they could able to produce a calf from a oocyte retrieved from a frozen and then thawed ovarian tissue. So that has encouraged many researchers to go for this kind of technique to improve the quality of oocytes from the part of the ovarian tissue that can be frozen. So cryopreservation and recovery and subsequent culturing of oocytes will help in preserving endangered species and also to maintain biodiversity. Why pre-enteral follicles? Because they are the source. Around 90% of the oocytes can be obtained from there, and it forms something like ovarian reserve of oocytes. Then other advantages are oocytes in the pre-enteral follicles are less differentiated and almost metabolically inactive, making them more resistant to damage during the storage, especially during cryopreservation. And one more thing is, genetically they are resistant because they are arrested in prophase rather than metaphase. Next slide, please. So, just to give a glimpse of types of preservation, initially, conventionally, they started with slow freezing, which has many disadvantages like formation of crystals and damage to cells or shrinkage of cells. So then they started rapid freezing, that we call it as vitrification. Vitrification needs high concentrations of cryoprotective agents, which needs to be standardized. So I mentioned this crystal formation can be inhibited through vitrification. Then other advantage with vitrification is by the cryoprotective agents. So this is a glimpse, and so many things are explained there. Next slide. So these are the cryoprotective agents that are going to be standardized in this study. So these are the different types of cryoprotective agents that are used. Mentioned types, permeating and non-permeating, and their role in the vitrification. Okay. So with this background, the study was formulated with this objective to evaluate the growth, viability and antral formation of pre-antral follicles obtained from fresh ovaries and from cryopreserved ovarian cortical tissues using varying concentration of cryoprotective agents. So experimental design is here. We have collected ovaries from the sheep from the abattoir, local abattoir. Then they are brought to laboratory, processed. So one set is processed for directly fresh cortical cells, pre-antral follicles, which are set for culture, and other part is prepared for vitrification. So we have done many parameters, but I am concentrating only on the viability and morphology of oocytes in this study. Next slide, please. So because these are the cryoprotective agents that is going to be needed to be standardized. So we used two treatment, VT1 treatment and VT2 treatment. So in VT1 treatment, again to standardize it, we have taken 3 samples with varying concentration of different cryoprotective agents. So next slide, please. So after the preservation for 7 days, so we retrieved that and then we have washed the samples with the different concentration of sucrose again to standardize it and then set for culturing of oocytes. Next slide. Next slide. Okay. Then these are the parameters, and parameters with this criteria we have done the result. Next slide, please. So these are the results. Growth rate. Fresh pre-enteral follicles showed a significantly higher growth rate compared to the frozen and retrieved follicles that we have done through treatments. And among the pre-enteral follicles in VT1 treatment and VT2 treatment, VT1 showed a better result than the VT2. Next. Then viability rate, fresh PF showed better viability rather than VT1, VT2. There was no significant difference between VT1 and VT2 treatments. Next slide, please. Antral formation rate was good in fresh pre-enteral follicles And pre-anti-conformation VT1 treatment group showed significantly higher anther formation than the VT2 treatment group. So conclusion, fresh pre-anter follicles are better than the one which has been frozen and thawed. But of course, always we can't go for fresh pre-anter follicle culture. So vitrification is better. Among the vitrification, VT1 treatment is better than the VT2 treatment. Thank you.
Thank you, doctor. Any question? So my question is, why when that fresh ovary yielded better result, then what is the basis of taking treatment 1 and 2, VT1 and 2? What is the basis? What was your hypothesis?
Sir, always fresh ovarian follicles is very difficult to get. So sometimes you need to go for vitrification so that in future you can get it retrieve back and sell. Especially animals which are good in breeding, high priced animals. And the cortical, ovarian cortical can be standardized for by biopsy technique, so that when animal is alive itself.
Do you find there is a scope of improvement in this cryopreserved ovaries that in terms of yield? There is a scope?
Yes sir, definitely. There will be culture, if you standardize it better with the media and freezing technique, definitely you can culture, the yield will be more.
Okay, thank you. Thank you.
Thank you sir. Thank you.
So next speaker is FTIR driven spectral biomarker. It is yours.
My presentation is the last one. OP8. I need to act as rapporteur in
Later. Last one is
expression of neuromodules. OP8.
okay, please.
A very good morning to all. I am here to present on the topic expression of uromodulin in urine of buffaloes at the estrus and diestrus stage of estrus cycle. Timely estrus detection is very important for improving reproductive efficiency in buffaloes. Next slide. Constraints in detecting estrus in buffalo being the poor manifestation of estrus signs, silent heat, heat stress associated. Modulin would have a physiological role around the estrus phase in buffaloes. Therefore, the present study was framed with the objective, next slide, to quantify the expression level of uromodulin in urine of buffaloes at the estrus and diestrus stages of estrus cycle by Western blot. So these were the steps involved. Cycling Murrah buffaloes, which were maintained in a private farm, were grouped into the diestrus phase animals were grouped as a control group, and animals in the estrus phase which showed the overt signs of heat were grouped as G2, and the animals which showed poor signs of heat were grouped as G3. Blood and urine samples were collected from all three groups of animals. Blood samples were used for the estimation of estradiol and progesterone, and urine samples, the midstream urine samples, were then used for the study of quantification of uromodulin by Western blot. So in the Western blot, we have employed the use of polyclonal antibodies raised against uromodulin in sheep in the study, and the expression levels were compared by using one-way analysis of variance. Next slide. So these were the slides showed. We have used a combinatorial approach for identifying the animals at the estrus phase, so as to group them as regular estrus and silent heat animals. And the slide shows a typical fun pattern of saliva as well as cervico-vaginal mucus. And in the case of animals in the estrus phase, both regular estrus and silent estrus, the estradiol levels were significantly higher when compared to the diestrus phase, while the serum progesterone was significantly lower in the animals in the estrus phase when compared to the diestrus phase. Next slide. So this slide shows the expression levels of uromodulin in the three groups of animals. The levels are highest in the regular estrus group, followed by the silent estrus and then the diestrus animals, though the levels are found to be non-significant. Next slide. And uromodulin is found to form viscous barriers in the oviducts under the low physiological pH that prevails during the estrus phase to prevent the infection. In cattle, the mRNA transcripts of lactoferrin were highly expressed in the uterine tissue, and also the protein was highly expressed in the bovine cervicovaginal mucus during the estrus phase. In this context, uromodelin was found to enhance the polymorphonuclear phagocytosis by means of binding to the lactoferrin protein, and also the role of uromodelin around the estrus phase in buffaloes would be to prevent the rise in the levels of bacteria beyond physiological levels. Next slide. Therefore, the conclusions can be drawn saying that the increased expression of uromodelin at the estrus stage would be to have the antimicrobial role. The study also implicates the role of estrogen levels and the uromodelin expression. High ROS levels might attribute to the increased uromodelin levels. Next slide. And a simple method has been developed. In fact, this is the first study in which has standardized a Western blot for the identification of protein levels in urine in buffaloes, and also the suitability of commercially available polyclonal antibodies for further proteomic research in buffaloes. This is the outcome from the study.
Okay, thank you, Manasa. Any question from the audience?
What is uromodelin?
Uromodelin is a glycoprotein which is produced by the normal tubular cells of the kidney in all the mammals.
Molecular weight?
Molecular weight, it ranges from 65 to 110 kilodaltons based upon the glycosylation.
Okay, thank you, Manasa.
Thank you.
Thank you. So next will be Koslish Ranjan. Okay, please. She will be at the last.
Very good morning to all. My topic of presentation is complete genome sequence based comprehensive analysis of C. origin Bluetongue virus serotype 1 from India. So next, virus caused by the Bluetongue disease caused by the Bluetongue virus and occurs in domestic and wild ruminants, especially in case of the sheep, but also in goat, cattle, and camel, and is transmitted by the Pulcoides vector, that is also called biting midges. So it is characterized by the high temperature, cyanosis, edema of face, swelling of only band, and then next. Coming to Bluetongue virus, so this virus has 10 segmented double RNA segment. It has double-celled non-enveloped cytosymmetry and 7 structural and 5 non-structural proteins. So here the B2 protein is a serotype specific proteins. So till date 28 different serotypes have been isolated from the world and 23 from the India. Next. Objective of our study is the complete genome sequence based molecular characterizations of Bluetongue virus from India. Next. So we have collected the virus isolate from the Andhra Pradesh in the 20 cell line adapted. Next. So we have isolated the viral nucleic acid with the help of the standard triazole method, and there is RNA there. They are out for the 1.5% agrogel electrophoresis. Next. Then we have done the RT-PCR for cDNA preparations of the virus sequence. Next. Then NS1 and BP2 gene based PCR we have done. So NS1 gene PCR was done for the confirmation of the sample as the BTV, and all the 24 different serotypes primers were used for the confirmation of the serotype of the virus as per the standard protocols. Then complete genome sequencing, the metagenomic study also we have done using ion tolerant machine with the help of the 400 vesicular sequencing chemistry. Next. And bioinformatics study we have done. So we have done the cell genomics workbench for the sequence assembly of the metagenomics work, then BLAST and BioDeep and MegaX and sequence demarcation tool were used for the phylogenetic study. So this is the result of this cell culture grown of the virus. So virus is clear cut showing the fomic degeneration, detachment from the cell surface. It is cytophatic effect. Next. The 1.5% agrogel electrophoresis shows the 10 different segments of the BTV. That is specific features of the BTV in 3:3:1 migration pattern. Next. NS1 gene based PCR shows 366 base pair amplicon that is specific to the BTV, and among the all 24 different serotypes, only BTV serotype 1 has shown the result. Remaining all 23 serotypes didn't show any result. Next. So genome assembly are done using the CLC genomics workbench, and we got after de novo assembly 1018 contigs with minimum length of 548 nucleotides at 25x coverage, and mapping was done using the standard BTV1 segments from the NCI database. Next. So this is the genomic characteristics of the all the 10 different segments of the our isolate. So these segments were submitted to the gene bank sequence with different session numbers. So our segment 2 shows maximum identity only with the BTV serotype 2 of the gene bank database. That's confirmed that is serotype 1 only. Next. Now coming to phylogeny and sequence identity matrix of all the 10 different serotypes. So all the 10 segments, segment 1 showing maximum nucleotide and amino acid identity with BTV serotype 9 from the India and placed under Eastern topotype. Then segment 2 showing maximum identity with BTV1 from India and China and placed under Eastern topotype viruses only at Eastern origin segments. Next. Now, segment 3 also showing maximum identity with BTV 1, 2, 9 and 21 from Indian isolates and placed under Eastern topotype viruses. Now, segment 4 showing maximum identity with serotype 1, 2 and 16 from India and placed under Eastern topotype. Next. So here segment 5 showing maximum identity with BTV serotype 1, 5 and 9 from India and from Western topotype. That's the typical thing here. See, placed under Western topotype. And segment 6 also placed under Eastern topotype and maximum identity with BT1 from India, Greece and China. And segment 7 shows maximum identity with BTV1 and placed under Eastern topotype. Similarly, segment 8 is also of Eastern topotype origin and placed with BTV1 and 16. Next. So segment 9 shows maximum identity of 97% BTV1 from India and placed under Eastern topotype. So segment 10 also showed maximum identity with BTV1 and 16 and placed under Eastern topotype only. So in summary, we can say complete genome sequence of our virus confirmed that it is BTV serotype 1, and this isolate is a reassorted isolate which has all 9 segments of Eastern topotype and segment 5 is of Western topotype. And previous study has shown that these reassorted viruses they cause much more pathogenicity and severe diseases in the animals.
Okay, thank you. And it's open for any question. What is the size of the cDNA?
cDNA size actually is vary on the all different segments. So segment 1 is size of 3.9 kb, so CD will be 3.9 kb. And
No,
segment.
no, I would like to understand that if the whole cDNA,
Yes.
it is not very big.
Yes,
The
sir.
segmentation was not required. Is it
No,
so?
segmentation I have not done, sir. This is the features of the virus itself, sir. It has 10 different segments. Naturally, sir. We have not done segmentation on our own, sir.
To segment, you mean to say part of the cDNA or?
So actually, sir,
different
this
strand?
virus is a Rioviridae family virus. And Rioviridae family have segmented genome as a feature. So it has 10 different segments like gluten virus, rotavirus. They have 11 segments. So it has 10 segments.
Okay.
Yes.
Thank you. Thank you.
Actually, you're going for the whole genome sequence.
Yes, sir.
So you got the sequence data. Whether you go for the sequence, the sequence studies, what are the changes of this thing will be there. So suppose there are some changes in the. Suppose you are getting some serotypes which are prevalent in India and
Yes, sir.
substandard. Maximum you are getting.
Yes, sir.
One probably in European and rest all are Asians, I think.
Yes, sir. Eastern nature, sir.
Okay. So whether you, what is the, actually how you can say that this will be here? Certainly when you are going some data, a lot of ATGC, so there will be some changes between this thing, whether you study this thing or just for the phylogenetic analysis you are telling this
Yeah, phylogenetic study and protein sequencing, we have reduced the protein sequence also, sir. So we have shown phylogeny only based on nucleotide here. So amino acid sequences, phylogeny is almost similar nature we are showing, sir. That's why shown only the phylogeny based on nucleotide, sir. Otherwise it becomes very deep presentation, sir.
Okay.
So results are almost similar.
No, it's okay.
Yes, sir. Based on protein also.
Okay, thank you. So next speaker is Labuddha Naveen. Anil to present.
Thank you, sir. Very good afternoon, very good morning to everyone. Basically, we all are for the betterment of the animal health. And the diagnostic is one of the most important part for the animal health. So we try to develop a neural network for the non-invasive tumor diagnostics. So here I am Dr. Anil Gatani, Associate Professor at Biotechnology Veterinary College, and this work is along with the Dr. Anita Munura. She is a mathematician, and all the neural network part was taken up by her, and it is based on the MATLAB work. Next. So canine cancer is a major veterinary health concern to which we have to diagnose early. And early and non-invasive detection is crucial for the diagnosis. For that we had taken the FTIR spectrum of the serum sample, and then we developed the neural network according to the wave numbers of the spectrum. Next. For this, we have these objectives. We have to identify the spectral biomarkers for the tumor specific, and we to develop the ANN network and to find the accuracy and the ROC curves for the neural network. For that, we took the total 40 serum samples. Among them, 10 is from the healthy, and we take the three tumors which are most common in the cancers at our clinics, that is the hepatic, vaginal and cutaneous. And we took the spectra at the ADR FTIR machine. The whole 400 to 4000 centimeter we had taken the spectra. And then these ADR we have pre-processed and baseline corrected, and then we developed the neural network. Next. These key bands which are specific for the serum, the amide 1, amide 2, the lipid, phosphate and the carbohydrate. These are the spectral bands where the specific changes may occur. Next. The neural network, we had taken the feed-forward type of neural network. For this, the tenfold cross-validation and the 4 classes we have taken. Next. These were the hidden layers that we had started from 500 to output layer is 3. Next. So this is the FTIR spectrum. Here we did not show the full spectra. It is from 800 to 1800. The rest is because we have some interesting feature in that area. So we are not disclosing that. So we took these spectra. Next. Then we mathematically derived the second derivative of these spectra. Next. And then we also took the second difference between the second derivatives of these spectra. Next. On the basis of these, we have for the ANN feed-forward neural network, we have to pre-process the data. So on the basis of PCA scores, PCA loading and the FDA, LDA, there are 2-3 more mathematical modelings were taken up, and from there we took the we did the pre-processing for the data, and then next, and we developed the confusion matrix and the ROC curve, and it shows very good specificity, sensitivity that the neural network was working properly. For that, the 70% data was used for the training purpose, and rest 30% data is used for the checking of these confusion matrix in the ROC curves. Next. So the T1 metabolism, it alters the protein and lipid metabolism and the carbohydrate also. The distinct biochemical variation, it was observed among the different tumor types, and neural network, it achieves more than 91% classification accuracy, that we can differentiate the type of these tumors with the healthy one. Next. So the FTIR, which is linked with the neural network classification, it offers the reliable, cost-effective and non-invasive tumor classification and diagnostic approach, and it can produce the early detection and classification possible. Thank you.
Thank you, Anil. We have finished well in time, so it's open for discussion. Okay, thank you.
Thank you.
Yeah. Okay, thank you.
Thank you all. Now we have this was the pending session for yesterday. Just we want to close this session, so there are some formalities. So I would like to. I think the lead paper presenters that those who have presented yesterday are not present today. If anybody is there, then I will like to felicitate our chairman. So. I'd like to request Dr. Anita Ganguly, Regional Director, Lala Lajpat University of Animal and Veterinary Science, Research, to come over the dais to felicitate our chairman. And I would also like to invite Professor Shatta Sharma from Assam Agriculture University, Khanapara, to felicitate. Yes, in that sir, please. I request Anita ma'am to felicitate sir. To please deliver to Emeritus Professor of our university, Professor S.N. Dev sir. He was also Director of NRCR and also Principal Scientist in IVR and my teacher too. Thank you. Now I request Anita ma'am please felicitate our rapporteur. Thank you, ma'am. So this marks the end of the technical session 1. So now we are going to proceed for the next session.
टाइमिंग ये सेट करेंगे पहले डिस्कस करेंगे
Let us start the Young Scientist Award program also. But I would like to invite the chairperson, Professor Shatta Sharma, to come over the dais. Professor Vijay Pandey and Dr. Anita Ganguly to be the chairperson of this session.
वो इन्होंने भी दिए थे जो वहाँ नहीं है सीनियर या फिर सोसाइटी एंड अगर सोसाइटी वगैरह भी वो भी या नेक्स्ट मंथ
বল বল বল
I would like to invite
ভাইয়া বাইরে আছে খাচ্ছে আমি বলছি আসি
Dr. Kavita Rawat.
আমাদেরটা হয়েছে ইয়ং সায়েন্টিস্ট শুরু হবে, তাদের লিস্টও নেই তো এদের কাছে। আচ্ছা, এখানে কত সময় পার স্পিকার? না না, তিনজন এসছে। আরে এটাই তো 10টায় শুরু হওয়ার কথা ছিল। হ্যাঁ, আমার আছে, তারপরে লিস্টটা তো দিতে হবে।
मैं बता देता हूँ। चल रहा है क्या? वाला खत्म हो गया, प्रेजेंटेशन खत्म हो गया, सेशन चल रहा है, वो जो नेट चल रहा है। हाँ जी सर, कहाँ है आप जा रहे हैं? ढूंढ रहा हूँ आपको। उधर कहाँ कौन सा सेशन? पोस्टर चलाया चल रहा है वो।
the prestigious award, Young Scientist Award. So we are going to start. So we have 3 young scientists to be presenting. The time allotted will be 12 minutes and last 3 minutes for discussion. So we can start one by one. First one is Dr. Biotechnology.
Good morning, respected chairperson, respected rapporteurs, and the delegates invited from all over the India, my dear colleagues, dear friends. So, myself, Dr. Partha Sarathi Behera. Today, here I am present for the research work for the Young Scientist Award. So, I am presently working as assistant professor in Department of Veterinary Biochemistry, Central College of Veterinary Science and Technology, Central Agricultural University, Imphal. I will next, please. So these are some of my credentials. I completed my India Master's from India, PhD from IUI. Next, please. Next. So these are some of the externally funded projects that I am involved in, funded by Department of Science Technology, Government of India, and Department of Biotechnology, Government of India. So the latest one just concluded in the March 2025 year, supported by the DST, Government of India. Next. So these are some of the intramural research projects that I am involved in. This is funded by our Central Agricultural University itself. So 6 projects in the intramural research project I have worked as investigator. Next, please. So this is the area, sir, I am working for the last 13 years in this genome recording approach, starting from my PhD at IVRI under the guidance of Asian National Professor Bhaskar Sama, sir. This genome recording is very interesting topic, sir. Here, as per the definition, changing the existing codons with synonymous rare codons without altering the amino acid sequence, that is the genome recording. It's one of the viable approach in the attenuation of the microbes. And this is particularly used for the customization of the target gene expression. You can reduce the expression by this genome recording. Next, please. So, actually, there are different properties, several properties of the genetic code like universality, non-ambiguous, non-overlapping, commaless, etc., including redundancy. So, we are exploiting in this genome recording approach the redundancy in genetic code approach to recode the gene. We are recoding the gene so as to reduce its expression. So, it can produce a protein which is having identical amino acid sequence as that to the original wild type. So, there is no difference in the amino acid sequence, only we are changing the nucleotide sequence by genome recording approach. So this has been used in making large-scale synonymous changes in the genome of many viruses. For example, respiratory syncytial virus, immunodeficiency virus, SARS-CoV virus, polio virus. Lot of virus the work has been conducted, and some bacteria like Streptococcus pneumonia. This approach has been utilized, as well as in one of my senior. I was involved there in Pachyderma multocida, and I am working on Salmonella typhimurium. Next, please. So the genes targeted for this genome recording can be a toxin gene, can be a virulent gene, can be a metabolic gene, or a transcriptional regulator, or a metabolic enzyme set. Next. So this is the topic. First, we worked on this recording the FNR. Next, please. So this FNR is actually the anaerobic regulator of this bacteria. Actually, during the bacterial passage through the gastrointestinal tract, it encounters number of adverse environmental conditions such as the metabolites released by commensal bacteria, increased acidic conditions in the stomach, including the low oxygen tension in the intestine. To adapt to this low oxygen tensions are actually, the FNR plays very important role in helping the Salmonella typhimurium to adapt to this low oxygen environment of the GI tract. So, what we did, sir, we record this FNR, which is controlling the metabolism of the bacteria in the anaerobic condition. This is the approach, sir, how we proceeded. Next. So then we conducted different parameters of pathogenicity, sir. So in the approach, just in the previous slide, please go to the previous slide. Just to the previous slide here, sir. The approach is that the recorded gene was first cloned in a donor plasmid PKSI1, and it was co-transferred
by electroporation into the bacterial genome along with the helper plasmid period take 1. The helper plasmid helps in the recombination process between this donor recombinant plasmid and the genome so that the from the original genome this decoded gene replaces the normal gene and the recorded mutant is produced. This is the approach that I have proceeded. Next please. Then we conducted different pathogenicity parameters to evaluate the pathogenicity of this bacteria like in vitro growth kinetics, growth competition assay, biofilm formation assay, macrophage survival assay, then in vivo correlation study, then fecal setting. So see this is the slide shows how we conducted the growth kinetics assay. Both wild and mutant bacteria are grown together overnight. From the overnight culture, it was subculture into the 50 ml LB process. Then from that, at every 1 and 2 hour interval, we took the alicot and taken the OD and plotted this graph. You can see here that in anaerobic condition, there is, though the difference was not that much significant, but there is difference between the wild and mutant bacteria. Next, please. So this slide shows the swimming motility ability of the wild and mutant bacteria. We tested in 0.3% LB agar plate without sodium chloride, and the bacteria wild and both were staked in the HEA plate and grown for 7 to 8 hours at 28 degrees Celsius there. The diameter of the migrating bacteria from the point of inoculation was measured as a turbid zone. We measured that diameter, sir. Then we plotted this graph, sir, in this to show that there is significant difference in the biofilm forming ability, swimming motility of the wild and mutant strain under anaerobic condition. Next, please. This is the biofilm formation ability of the wild and mutant strain, sir. Biofilm formation and motility both are intimately related, sir. And for biofilm formation, the motility is very utmost essential for the initial colonization and biofilm formation. That's why both are very intimately related and both are very strong indicators of the virulence of Salmonella typhimurium. So by recording this FNR gene, we have found that there is significant difference in the biofilm formation of the mutant as FNR recorded mutant in the under anaerobic condition as compared to the wild strain. Next, please. Next parameters I have conducted this macrophage survival assay. The monocyte derived from the monocyte derived macrophage from chicken were stimulated with LPS, then were infected with the mid-log phase bacteria in a multiplicity of infection of 1:50. Then, sir, after 24 hours and 48 hours of post-infection, then the bacteria lysed with 0.1% Tritonex 100, serially diluted and plated on HEA plate. So it showed that there is significant difference in the survival in the macrophage also. Next, please. So we also conducted the survival assay inside the raw 264.7 cell line of the macrophage. There also we got similar trend, there is significant difference of survival of bacteria in the mutant strain. Next, please. So colonization ability of the bacteria, mutant strain both to the liver as well as spleen also was significantly reduced in 5th day of post-infection. Actually, we infected bacteria with 4.5 mutant per CFU bacteria was infected, both wild and mutant bacteria. Then we found that intraperitoneal infected cell, and we collected the liver, spleen on 1st day, 3rd day, and 5th day post-infection. On 5th day, there was significant difference in the colonization ability of the mutant strain. Next, please. Mice also we conduct similar type of colonization study in the spleen here. Next, please. So this is the fecal setting, sir. Monitoring the fecal setting is also considered as an important indicator of virulence of Salmonella typhimurium, sir. We infected the bacteria with about 2 9 CFU bacteria, both wild and mutant bacteria were infected orally, sir, here. Then we conducted the fecal setting, collected the fecal on the 1st day, 3rd day, 5th day, 7th day, 14th day post-infection, sir. We can see here, sir, in the 5th day and 7th day, there is significant reduction in the fecal setting of the bacteria. So all this in vitro pathogenicity as well as in vivo pathogenicity study indicates that there is significant reduction in the genome, in the pathogenicity of the genome recorded mutant as compared to the wild strain. Next, please. Sir, after that, we conducted all the experimental, at the experiment levels, we conducted all the in vitro pathogen study. Then we proceeded to the preparation of another mutant from the single mutant which we called a double mutant. Next, please. So in that FNR mutant, sir, we recorded another gene, sir, that is ASVA. That is also another anaerobic regulator of this Salmonella typhimurium. We can see, sir, we have compared the study of both the single mutant with the double mutants, and single mutants are compared with the wild type. There is significant reduction, and the most interesting we observed here is that there is a synergistic effect in the reduction of the pathogenicity in case of double mutant. Next, sir. So this is the bioinforming ability. Here also we got the similar trend as compared to the other sites. Sir. Here there is also reduction in the pathogenicity synergistically, sir. Next. So this is the swimming motility study of both the single mutants and double mutant as compared to the wild strain, sir. We can appreciate here, sir, in case of double mutant, the sorbate zone produced is very less as compared to the single mutant and wild also. So this indicates a synergistic approach in the pathogenicity in case of genome recording. Next, please. This is the survival curve, the in vivo survival curve of the single and double mutants, sir. The percentage survival in case of double mutant were definitely less as compared to the single mutant. And here also we observed one of the synergistic approach in the pathogenicity in case of recorded mutant as compared to the wild strain. Next, please. Sir, after conducting all the in vitro in vivo pathogenicity, we went for this proteomic study. Next, please. We can see here we compared the proteomics of the wild and delta FNR mutants. Sir. We found that FNR regulates this TCA cycle, respiratory chain complex and ethanol metabolism, also controls the pyruvate metabolism, glycerol metabolism. Next, please. We also validated our proteomics data by real-time PCR, sir. We also got the similar trend, except in case of ethanol metabolism. That reason may be due to the post-transcription modification. Next, please. We also conducted the proteomic analysis of the recorded mutant. Next, please. We can see here. Next, please. Next, please. Here, sir, you can see that FNR first time we revealed that it controls the glycolysis, chemotaxis, and nitrogen metabolism in salmonella typhimurium. Next, please. These are the some of the classification of by different Panther database. Next, please. Next. 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 Sir, these are the some of the activity of this SSR activity of our project, where we have conducted a research facility training for the master and PhD scholars as per the mandate of the DST. Next, please. This is the exposure visit of the school students under the DST SAR project, where we visited the school students, our research facility, other facility. I am concluding, sir. So the target gene customization by synthetic genome recording using biased codon is a viable strategy for attenuating vaccinal virulence. Further increasing the extent of recording decreases the pathogenicity further progressively, and as I shown the data, it has a synergistic effect. First, robust proteomic evidence of our genome recorded mutant suggests or reinforces the role of this FNR in regulating the bacterial metabolism and virulence during anaerobiosis, and supports our in vitro pathogenicity study earlier. Thus, genome recording can be exploited to customize pathogenicity to a level which is appropriate for use as a vaccine, and it provides a new avenue for vaccine development. Next, please. Thank you, sir. These are some of the publications. I acknowledge the Department of Biotechnology, DST, Vice Chancellor CAU. I share it from National Professor, my guide, Professor Bhaskar Kumar, Dr. Ajay Kumar, my collaborator. Thank you so much, sir.
Thank you, Pato. Now, his presentation is open for discussion. So, if there is no any point to be discussed, so thank you.
Good morning to all, respected chairman, co-chairman, and rapporteur, and respected faculty. Myself, Dr. Prabhas Bajan Sir. I am working as assistant professor, Department of Veterinary Biochemistry, and I did my PG and PhD from IRA. Recently, I got a project from CCRS, that is Central Council of Research on Homeopathy. It is molecular evaluation of anti-cancer activity of combined homeopathic preparation of sugar and carbolic acid against canine mammary tumor, protein inhibitor, and in vitro model. Yes. So this is a project when we carried out from 2025 to 2028 in the collaboration of veterinary surgery and veterinary pathology community. So you know, canine mammary tumors are important tumors in dogs. Around 36% is the prevalence rate, and the canine malignancy is 41 to 53%, and the canine mammary tumor is prevalence more than the human breast cancer. And you know the clinical science and next. So these are the therapy available nowadays for the canine mammary tumor, permanent therapy, then radiotherapy, then surgical intervention, then chemotherapy, but all the therapies produce some undesirable side effects. And the surgical intervention only for the localized tumor mass. So there is no treatment for the metastasis analysis. So next alternative is complementary and alternative medicine, that is homeopathy. Yes. So we will target for giving puja and carbolic acid combinedly to the animal. So puja, what it does, it is releases the reactive oxygen species which activates the synthetic nitrogen activated protein kinase, which can activate P53, which is the tumor suppressor gene, and it releases its cytosol and activates capsid, which induces the apoptosis of the cell. And carbolic acid is one of the causes corrosion and destruction of the tissue. That's why the carbonated mammary tumors will be degraded. Yes. So this is our question and hypothesis, how the combined effect of this puja and carbolic acid show the anti-cancer activity. And we have two objectives to evaluate the anti-cancer activity of both puja and carbolic acid, so in vitro and in vivo models, and to understand what is the molecular mechanism behind it. Yes. So in vitro model, we will first collect the tissue, then cell culture, then we will group into 6. One is normal canine tumor tissue, then with positive homeopathic drugs, then with puja, then with carbolic acid and both. Then we will go for the molecular expression study and protein expression study. Yes. After doing the in vitro, we will go for the in vivo study. The clinical cases coming to the PBCC, we will group and we will first measure the size of the canine tumor. If it is more than 2 centimeters, then we will deblock it. If it is less than 2 centimeters, then we will give the homeopathic drug for 7 weeks. Then after that, we will take the post-treatment evaluation process. So this is the preliminary work we did. The Ki67 gene significantly decreased the cells giving both carbolic acid and puja. Similarly, the PCNA proliferating cell nuclear antigen also decreases significantly in the cells treated with both puja and carbolic acid. This is the antioxidant parameter we did. This glutathione peroxidase also significantly decreased in the cells treated with both puja and carbolic acid, and catalytic activity also decreases. Yes. So this project will develop an alternative of the effective homeopathic preparation against canine tumor, and this work also provides detailed mechanism how this works. Yes. So this is my PhD. Last year I finished my PhD at IVRI. We were able to publish this paper. So this is also the narrow gene is the anaerobic regulator of salmonella, and we check how the narrow gene modulates the lipid biogenesis. That's why we developed white Salmonella typhimurium and complementary strain of Salmonella typhimurium, and we give the treatment with the both, and we quantify the lipid droplet. And here it's seen that the wild type Salmonella typhimurium increases the lipid droplet accumulation at 6 hours, and the mutant type increases the quantification of the lipid droplet at 24 hours, and it was validated by the confocal microscopy and flow cytometry. Yes. Another work also we did for the protein-protein interaction of narel in Salmonella typhimurium. So we targeted 4 proteins: narel, nargine, SSRB, and FNR protein. We clone and express all the proteins in the prokaryotic expression system, and we show the interaction between different narel with nargine, narel with FNR, and narel with SSRB for different techniques. And it shows that the narel-nargine interaction is stronger than the narel-FNR and narel-SSRB interaction, and it was checked by the isothermal titration calorimetry, and it was shown that narel-nargine interaction is exothermic reaction than narel-SSRB, which is endothermic reaction. And also we got able to publish in current microbiology this work recently. Yes. This is my student research. We synthesize both green and chemical synthesized silver nanoparticles and check the antimicrobial effect against the Staphylococcus aureus and E. coli in bovine mastitis. And we characterize all techniques by transmission electron microscopy. This has found around 15 to 20 nanometer, and the zeta potential found around -33. And the antimicrobial effects show the green synthesize shows higher antibiotic effect than the
chemical synthesis, irrespective of the organism. And also we could able to publish this paper recently 1 month before. So this is my reference. Yes. Yes. Thank you.
Thank you. Now his presentation is open for discussion. Any point to be discussed?
Dr. Prabhas, one question I have that when you have synthesized these nanoparticles, the size dimension it is up to what?
The size dimensions that you have revealed with the FDSA?
Up to 20 nanometer.
Up to 20 nanometers. And the plant it is?
Plant is citrus liver.
Okay.
Citrus. We extracted the citrus and utilize for the green synthesis nanoparticles.
And LCMS it is also been done?
Yes. GCMS.
GCMS.
Yes. Citric acid found the major compound in the citrus liver.
Thank you for your presentation. But I need to know something from enrich my knowledge. Your group of animals, rabbit or dog? Dog. Because memory tumor is very frequent in dog. Very frequent. May I know one thing? Do you know the epidermal thickness of the epidermal layer of the dog? Do you know the thickness of the epidermal layer of the dog?
Yes, I don't know, but the myopicals.
Because you are applying carbonic acid, which is contraindicated. But you are applying. Therefore, I need to know the epithelial layer of the skin. Can you elaborate all the layers of the skin?
Sir, that's why we will dilute the carbon.
Can you elaborate all the layers of the skin? Epidermal layer of the skin? How many?
That's why we will dilute the carbon acid.
I don't know. I want to know the basic question. I am not a man of biochemistry. I am a man of medicine. Therefore, I do not know all these things. But I need to know from my own knowledge what are the layers of epidermal layers in the skin of dog. Because you are applying, therefore you must know the structure on which you are applying. Whether carbonic acid could be applied in the skin of dog or not. If it is not to be applied, then what I am going to do further research on that? You know this structure. I tell you for your information, the epidermal layer of the skin of the dog is 6 times thinner than human being. 6 times thinner than human being. So we are applying corticosteroid in a subject which is vulnerable. This is my one basic question to you. So if the basic question is not being fulfilled, then what is the findings and what is the all these parameters are being covered? Thank you.
Why have you presented 3 research here? You should have prepared one research or one either project or your one research of MSc student or PhD student. You have presented 3.
Sir, in my lifetime I did this, that's why I presented here. And recently I joined, I finished the PhD, that's why I and these 2 papers
It's my advice. When you come to for a competition like a senior scientist award or young scientist award, you come with a single project, with a title, with a hypothesis, with a objective, with a result. First was enough for presentation. Your first presentation was enough for the presentation. Do you have any homeopathic doctor as a SRF in your doctor?
yes, sir.
Thank you. Ajit Kumar.
Good morning, all. I am presenting here one of my project, just completed project, that we have developed the visual detection assay for three bacteria, E. coli, Streptococcus and Staphylococcus. Next please. And these three bacteria are involved in the causing agent of the mastitis, and most of the mastitis, most commonly you can say the mastitis caused by these three bacteria, E. coli, Streptococcus and Staphylococcus. And most of the techniques, older techniques, we have to culture the bacteria for identification of that bacteria, and that requires much time. Then the next nucleic acid-based techniques are there. That techniques give you the accuracy, but that also require expert person. So there now world is going for some biosensing field site technical field site test format. So we have this biosensing of the three bacteria. Next please. Next. So for that we have used full nanoparticle for attaching the probe. One probe design, and that probe is attached to this gold nanoparticle, because gold nanoparticle having good surface to volume. That's why any kind of the probe can be easily attached there, and there is a very easily in most of the cases in biosensing gold nanoparticle is used. Next please. So our objective was to detect the E. coli, Streptococcus and Staphylococcus from isolated from milk sample by optical biosensing using oligonucleotide. Please, next please. First of all, identify the bacteria by using the primer, a specific primer. We use the standard bacterial standard in all three cases, and then we have selected one gene for that particular probe designing, and to confirm that by three genes are there. Top gene is for Streptococcus, then the gene is for Staphylococcus, and female is for E. coli. And then the probe is designed, and that custom synthesized from the vendor, and then we synthesize the gold nanoparticle by citrate method, most commonly method is there, and then this gold nanoparticle conjugated with this probe, and then characterized with the spectrophotometry, then the SEM, TEM and AFM. Then finally we optimize the condition for the biosensing. Next please. This is your first PCR confirmation of the all three bacteria, Streptococcus aureus, and in case of the Staphylococcus aureus, Streptococcus eclexia and E. coli. Next please. The characterization of gold nanoparticles is in spectrophotometry. The probe, after probe conjugation, there is a shift in lambda max that indicate that the probe is conjugated, and then we perform the SEM and TEM, and that indicate that all nanoparticles, they are not lose any property after the attachment of this probe. So all these data is showing. Next please. Then probe is actually another test is the hybridization assay. We took just complementary DNA, whatever probe we designed, and we performed the hybridization assay, and finally we released the probe with the complementary DNA by using beta methylphenol, and run on the gel. We find that the one band is there that indicates that the probe is properly attached to the gold nanoparticles and it's working. It's hybridizing with the DNA. So for that initial extraction we use complementary to the probe. Next please. Then we perform the conjugation of the gold nanoparticle probe, and for that several steps are there. First we use TCF buffer, and then we add gold nanoparticle, then we have probe, and this is overnight protocol. And finally we conjugate the probe with the gold nanoparticle. Next please. The working principle of this is that in presence of we can use salt as well as acid. We use acid. So in presence of complementary DNA, after adding the salt or acid, there is no color change because the gold nanoparticles do not aggregate. If absence of this complementary DNA, gold nanoparticle will be aggregated and that will give blue color. So this is clear indication that your probe is combined with your hybridized with your target DNA or not. Next please. So we first identified that how much HCl is required for aggregation of the gold nanoparticle. We performed say first standardize the temperature and then we standardize the HCl concentration. Next please. We find that after 3 microliter, in each 1 microliter, 1 microliter, 2 microliter, 3 microliter HCl is added. So we find that after adding 3 microliter of the HCl is for all 3 bacteria, it may be vary by presenting one. Okay. So it is some bacteria is 1 in 2 or 3 microliter adding HCl. DNA is that full antibody aggregating. So that is the HCl concentration for the processing experiment. Next please. So we isolated the genome. First we start with the genomic DNA. We isolated the genomic DNA from bacteria and then perform the same hybridization experiment. And next please. And we found that that after 3-4 dilation, we are able to detect 1.2 nanogram per ml of DNA in case of Streptococcus aureus. Next please. In case of Streptococcus glaucia, we are able to detect 8.56 nanogram per ml of DNA. Next please. With E. coli, we are able to detect 9.94 nanogram of DNA per ml. Next please. Then we perform with the culture. After isolation of bacteria, directly using culture of the bacteria. From in that we have detected bacteria that we. Next please. So first of all we culture the bacteria and then we simply lyse the bacteria by natural method and that is used as a sample and we detected in that. Next please. So we are able to detect up to 360 CFU per ml of bacteria in case of Streptococcus glaucia culture. Next please. Then we perform spike study in milk with Streptococcus glaucia. We added this Streptococcus in milk, soil milk, and then we culture. After that, the same method we perform step lysis method, and then we perform and that we get the 600 CFU. We can able to detect 600 CFU per ml of bacteria. And same with the confirmed with the PCR. We perform compare with the PCR also. Next please. Next please. Then we also use specificity assay. We use whatever bacteria we have in our lab. So we find that the Streptococcus glaucia giving the red color that means positive, and all other bacteria giving the blue color that is negative. So this method is have specificity. Next please. Then the E. coli culture, we are able to detect up to 1.2, some 1.2 to 10 to power 1 CA2 per ml. Next please. Then the specificity assay of E. coli with the different bacteria. In case also, only E. coli giving the positive result, other bacteria not giving the positive result. Next please. Then we have validated the directly in case of E. coli. We are able to directly take the milk sample from clinical mastitis testing and we validated in 13 samples. In all samples, we got the positive result. Next please. Then the specific assay of asparagus assay with different bacteria, we perform with different only Streptococcus aureus give the positive result, other bacteria negative result. Thank you. So, this is our project work.
Thank you. Any point to be discussed
What is the novelty of
on
this
his
work?
presentation?
What is the novelty of this work?
Novelty is we have our own probes. So this is novelty you can say, and methods are also, sir. Yeah, actually, everyone this kind of work is already performed. You cannot say that I am only person is doing this.
So then what is the purpose of your doing here?
Its purpose is doing here, sir. What we are doing that we are going for some lateral flow assay for that. Sir.
So that you are present all your data when you tomorrow you are preparing something and it is coming to IPR issue, you are already disclosed. You cannot use it this for IPR issue.
No, sir.
Then why you are present here?
Actually, sir, I have also published.
Then you cannot go to IPR.
I know, sir. Actually, sir, I don't have idea. That's why.
No, this much you are doing hard work. Why you are not going to filing patent or something else? Why you are publishing here? Why you are presenting here?
Actually, he has presented, he has published the work.
I published that.
He cannot go for the IPR.
Again, why you publish it? Then why you are coming to presentation here? That is also you have to before publishing. You have to present. That will be innovative idea, innovative things. Already it is a public domain your work.
Yes, sir.
Then coming here, that means what is useful?
Actually, he is presenting
His work.
as for young scientists.
Still, it is already public domain. The information is there. So new idea only present that will be useful for all the scientists, all the delegates. That is my idea. My means suggestion. Up to you. You can decide.
Actually, it is not clear from the society that what should be presented in the young scientists report.
Okay, sir.
So that's why the young scientists, these competitors are not knowing what to be presented. Somebody is presenting 3 works in a same presentation. Somebody is presenting this combinedly a 5 project in a same presentation, and he is presenting something that is already.
He already associate professor. He already assistant professor. They are not small young people. They are not completed just MVC, PhD. They are already in the university. They know all these things. Without knowing this presentation means then how will you utilize your knowledge in the IPR? How will you get the pattern? How your performance should be in the revealed outside?
Sir, we are assistant professor, not scientists actually. We are doing some science, but my main motto is teaching, sir. And our university is not promoting that kind of things. If I am going for that IPR, this will be my मेरे लिए बहुत ज़्यादा वर्क है उस तरफ कुछ जानता ही नहीं कि IPR कैसे करते हैं ऐसी जगह से.
No. What I am telling you are doing work. You are doing work. Very good work.
You can personally meet him and discuss.
No. You are doing very good work. I am not telling. You go to IPR. I am telling.
I know, sir, but I tried, sir, but no one is helping. That's the problem, sir.
Sir, then you come here. I will help you.
Okay, sir. Okay. I have other works also, sir. That can be have IPR. I have 2-3 works.
With the permission of the chair, I am not going in jugglery because I know little about that. I want to ask some basic question, which is actually what are science, what are scientific derivations, what are scientific knowledge we are uplifting, that is what we need to, but it has to be extended. Ultimately, it has to be extended to the field life, so that the animals are benefited, farmers are benefited, human populations are benefited. My question is this: you have taken 3 bacteria, Streptococcus, Staphylococcus, and E. coli. Can you please tell me which causes death within 24 to 48 hours? Which bacteria causes death within 48 to 72 hours? Among them may be, they may be when order may be completely damaged. And then high rise of temperature, where sometimes fluid is needed.
As far as my knowledge, sir, in acute cases, Streptococcus is a main bacteria. In acute cases, and in most of the cases, you can find the Streptococcus. And clinical, sir, E. coli is confirmed. In every we get the E. coli. All clinical cases. Clinical mistake.
I tell you, E. coli is the main. E. coli, it affects the stuffing of the organs. And there will be septicemia. But in other cases, only local treatment may do. But here the local treatment will not be.
Sir, actually, I have another project in that, sir. doing same delivering the drug with the help of this method. And that I able to deliver the drug. Whatever inflammatory therapy is available, apart from that, sir, we have more efficient delivery. And that passes this barrier, this big barrier is there, that drug able to deliver there.
Thank you. We are paucity of time. So, thank you, sir. Okay. Now I request Sonali Dwarkar to present her thesis for Best Thesis Award.
You asked me anyway. Not already we have filed. Somebody will be
So good morning. So good morning, the eminent scientists, respected chairperson, respected rapporteurs, innovators and researchers. Myself, Dr. Sonali Borkar from the Department of Veterinary Biochemistry, Nagpur Veterinary College, Nagpur, would like to equate you all on the research study that has been conducted as a doctoral research part on entitled as Studies on Synthesis, Characterization and Evaluation of Phytoengineered Silver Nanoparticles as Antimicrobial and Wound Healing Nanomedicine in Animals. Sir, as we know that many of the, from last few years, many of the researches, workshops and symposia, they have been conducted on the AMR, and recently on the UN platform, the Global High-Level Ministerial Committee, they have been contested that the AMR is not a distinct threat, but it is happening now. So conventional antibiotics, it is of least use. Now there is an urgent need and a high time for an alternative replacement for this process. And data also reveals that an alarming situation that the death toll will rise from 7 lakh to 10 millions in India. And also one of the status, it is that, next one, our data, it is in the way, and the research, it is on the way to have a pay back the conventional antibiotic, and that we should switch on to the green nanotechnology. Next one. So with the following three stringent objectives, we have undertaken the study, that is the phytofabrication and characterization of silver nanoparticles, and to evaluate the antimicrobial potential of phytoengineered silver nanoparticles, and to evaluate the silver nanogel in rat wound model, and lab-to-lab course, that is also it has been conducted in the clinical cases. Before the actual conduction of the PhD work, we have conducted a pilot study. In that, we have taken 4 distinct medicinal plants that are Azadirachta indica, Moringa oleifera, Platoria ternacea, and aloe vera. After that, we have phytocharacterized it, and after that, we have the result revealed that the Azadirachta indica have the potential to produce the stable nano-sized particles, and further we have conducted the research study. The technical program, it is vast. It is divided into 4 distinct phases. In first phase, we have phytochemically screen the plant extract, fabricate it, and then with the different characterization studies, we have conducted the characterization studies. And in next phase, we have done the in vitro antimicrobial, antioxidant of efficacy of silver nanoparticles and that of the aqueous extract also. In phase 3, we have dealt with the in vivo wound healing capacity of the silver nanogel that we have prepared in phase 3 in excision wound model in albino-vista rats, and in phase 4, that is most important, that the clinical trials has been conducted, that we have done the efficacy and potentiality of the phytofabricated silver nanogel in the form of the silver nanoparticles that we have applied in the clinical cases. First, in the first phase, I already emphasized that there is the preparation of the aqueous extract, and for that the 1 millimolar concentration of the silver nitrate solution has been used. Next one. So, with the standard protocol, standard procedures, we have prepared the silver aqueous extract. Then, next one. For the phytofabrication, we have done the HRLCMS and the FTR analysis of the aqueous leaf extract. Also, next one. We have done the AI-IGP synthesis where we have taken aqueous extract, 1 concentration and 9 parts we have taken of 1 millimolar silver nitrate, and then we have prepared the silver. This is our end product of the synthesized silver nanoparticle. Then it is further characterized, of course, by physical means we have gone for the synthesis. But what about the actual its applicability? So for that we have characterized this synthesized silver nanoparticle by using the UV spectrophotometry, FTIR, that is the Fourier transform infrared spectrophotometry, which has been done with the IIT Mumbai, FESM, it has been done with IIT Roorkee, and also the XRD analysis. In phase 2, we have done the antibacterial and antioxidant efficacy where I have done the MIC, protein leakage assay, and also the free radical scavenging assay, that is the DPPH assay I have been done. Next please. Yes, the broad dilution method, for that the MIC have been done for the E. coli and for the Staph aureus. Next one. For the MRSA have been done. Next one. In the phase 3, this is the important phase where we have prepared the silver nanogel by taking into consideration the carbopol powder, glycerol and all the contents in the specific ratio and the standardization of procedure, we have prepared the silver nanogel. Next please. For the experimental design for the in vivo study, we have taken the 5 groups. In the 5 groups, 30 distinct albino vista rats, they have been taken. Prior to that, of course, the ethical committee, institutional ethical committee permission have been taken. Next one. These are the experimental group where the negative control, only the application of the saline extract, it has been applied on the excision wound model. Positive control, the povidone-iodine, it has been applied. In the third group, we have applied the aqueous extract of Azadirachta indica. In the fourth group, only our required, our main purpose, silver nanoparticle hydrogel has been applied. And lastly, only the base it was applied. This is the excision wound model, about 1.5 centimeter diameter thickness. It has been excised on the vertical, either vertical side of the vertical column of the rat. Next please. Here it is how it has been done. Next please. The samples, they have been collected, blood samples, then bacterial isolates and the tissue homogenate, they have been collected for the further analysis. Next. Wound kinetics have been studied. Next please. Estimation of the tissue, connective tissue parameters, which are the indicators of the wound healing, that are the hydroxyproline, hexaluronate acid and hexosamine have also been estimated. Simultaneously, the MDA, GSS, SOD, catalase and quantitation of the protein in the granulation tissue have also been done. Next please. The DNA quantitation have been done in the tissue homogenate. Next one. Mild analysis have been done for both, for the in vivo bacterial isolates and also for the clinical samples also. So for both we have done the mild analysis. Next one. Yes, in the fourth phase, that what happened, our biochemistry relies only to lab, but this is the main target and main questionable for us, how it should go from lab to lab. So this is a risk for us, but we have conducted trials on the clinical cases, on the wounded, different wounded, we can say the samples in case of the clinics only. So many of the species we have taken. Nextly, next. Statical analysis is about, it has been done of SPES and other parameters. Next please. results. These are results. These are there. That we have conducted the phytochemical screening by HRLCMS. Please go on for. These are the meaning of the phytocompounds. They have been identified, which have the flavonoids, phenolic compounds, alkaloids compound. Then these are the negative and positive potential HRLCMS phytocompounds. They have been identified. Nextly, these are the important wound healing factors. Nextly, FTR has been conducted. Next one. From this, it is evident that from the light yellow color, the optical emission we get lastly at the dark brown color. So it is clearly indicate the synthesis. This is the importance of the SPR. This is the pronounced case where we get the SPR images at this 440 nanometer. This is the important one. Nextly, this is the second one we have done about the 55. But nextly, via permutation and combination and optimization of pH and temperature, we have achieved the size of 25 nanometer. Next one. FTR have been done also for the AGNPs. So it is clearly evident that the phytocompounds, they get encapsulated on the surface of the silver nanoparticles. We show the reduction and the stabilization of the nanocompounds. Nextly, X-ray diffraction. These are the strong peaks where we show the crystalline nature, that is FCCC nature of the nanoparticles. Nextly, yes, MIC has been done where we have got the good results on E. coli rather than the MRSA. Next one. Nextly. So DPPH analysis also shows the prominent effect of the AIGNPs. Next one. This is the same case where we have got the good results, that the IC50 we have got good in case of the AIGNP rather than of the aqueous extract, but of course it is less in as compared to the control. That's one. Wound contraction. Next. Wound kinetics, they have also been found, and in the this is a good picture where we can see that on the 12th day we have found the complete wound healing. So we are at the AIGNP group, which is our main achievement. Next one. There's oxidative stress markers that previously emphasized, that LPO, GSS, SOD and catalase have also been very good, have effect of the AIGNP. Next one. Biochemical parameters, hexuronic acid, hexuronamide and hydroxychloroquine, which are the collagen markers, they have also been conducted. Nextly. Yes, this is the DNA concentration, it is found high. Next one. These are the multi-top analysis. Please speak up fast. Where we have got both for the in vivo and for clinical samples. So bacterial isolates, they are found silver. This Staphylococcus caesuri I have found in case of the clinical samples, which is one of the wounded bacterial isolates which are present in the clinical samples. Next one. Yes, CRP and inflammatory markers, they are within the threshold level. Clinical sample, yes. Again. Yes, these are the pictures. We have found the good results at the end of the study. Here many of the lacerated, wounded, different type of ulcerative we can say the lesions, they are there, and they've been treated with the AIGNP gel. The last one, here it is the cases, we can see, and at the end we have found the good result. These are the blisters. Next one. Next, this is a feline case, this is umbilical, this is a again the umbilical wound, then this is a lacerated wound. Next one, complete wound healing, it has occurred. Next one. Conclusion, we can say at the end, I will conclude with my PhD research guide, which is guided, my guide, it is Dr. Manju Rahman. They have guided me, and finally I conclude with this, that AIGNP synthesized using aqueous azadirachta indica leaf extract with a range of 25 to 70 nanometer exhibited potent antimicrobial and antioxidant property effectively, which combats the major antimicrobial resistant pathogens that are MRSA and E. coli, and promoting enhanced wound healing in both in vivo and clinical cases. So this suggests that 0.02% of AIGNP hydrogel, it has been armamentarium, could be a promising agent for treating chronic and acute wound, which is one of the steps towards the antibiotic stewardship. Thank you. These are few of the we can say the scientific reports, and this before this it had also got patented. My PhD work, it have got patented right now. I am happy to share with you. Thank you, sir.
Thank you, Sonali, for your presentation. Now our presentation is open for discussion. Any point to be discussed? One or two points.
Which type of gel did you use for this?
Yes, sir. I have used this neem nano silver gel that I have
gel base.
Gel base carbopol plus.
Carbopol.
Yes.
And what was the pH?
pH it was
For the
7.2.
gel.
7.2.
7.2.
Yeah.
And what was the pH of your synthesis?
pH it was around, it was also 6.9 to 7.
Actually, for the silver, it is in the alkaline. If the silver have to be stabilized in the nanoparticle form, it has to be in the alkaline form. Otherwise, your particle size will be different and the stabilization is also affected.
But when we see with the FPSM and FTR, it has been there.
What was your stabilization up to which days you did check the stabilization of nanoparticles?
See, when we have prepared that particular assay, when we synthesize, we send to the lab. Okay. FPCM, every point it is not possible. We have standardized the protocol. That batch we have sent for FTR. That batch we have to send for FPSM. That batch we have to go for.
I'm not asking about that. What was the stability of your nanoparticle? Have you studied that? That up to
Stability.
30 that stability of your
I
nanoparticle.
have a gel compound. It has from 2 years, it have not any antibacterial effect, antimicrobial effect, no fungal accumulation it is there. That still I have with me. So that stability is there, and when I applied that one to the clinical samples.
I am asking about the synthesized nanoparticle, not the gel.
Okay.
What was the stability? Have you studied that time period of your stabilized nanoparticles?
That stability it shows in the clinical trials only. If it is not got stable, then that nanoparticles.
No, first you have to.
For the stability, I have done the FTI, I have done the XRD.
That is not the stability, no. That is your. You just prepare today or 5 days. You just send it for the characterization. Yes. You have to check the stability that up to how many days it is stable.
Then I have sent, whether have there any such any study, then let me know, sir. I will implement that. I will implement that because these are some of the factors that we have very much concerned. If it is shows the efficacy, then the proof
if it
there
is
with
stable, we have to tune the things. What was the size? If your size is different, it get accumulated and
agglomeration.
What you are telling that in the gel, if it is not stabilized in the preparations, then it will get also accumulated and also get agglomerated in the gel form also. Because your pH is also very much different.
But it is not so much.
Thank you, Vitali. Thank you.
Thank you, sir. Thank you, sir.
Sonali, you have done very good work during your PhD research work. My question is, by seeing your presentation, I was thinking that how much budget you have used in this project, and will it be economical for the farmers to use such kind of hydrogel for wounding?
When I have prepared this nanogel, I have also drawn the economics. Around 123 rupees that formulation can be done, or 50 gram.
Then it is okay. And how much budget you have used, just for my knowledge, for this project?
For the budget, it is too less, not so much. But for the characterization study that we have to go for the outsourcing, and that budget it is, when total budget I will get only apart from the characterization, it is only 10,000 or 20,000 only. Apart from characterization.
You have done very good work.
I have a question, if I can ask.
Yes.
Did you check? Did you?
Did you compare it to the commercially available products and you compared the study to the commercially available products?
Pure gel. It was there, which
Okay.
is only the silver gel only.
Okay.
That I have been compared.
Any antibiotic, classical antibiotic ointments kind of product you tried it?
No, sir. That antibiotics I have not been used
That-
because making the study we have some limitations. No. So that study we will do in later because again in the second phase I will do that. But
Because the work is
our
excellent.
purpose it is that it should be conducted in the clinical trials because you see in any of the papers they get restricted only to the in vivo models only.
Sure.
They have not been done in the clinical cases, and that
was our achievement, and we have found the good results of it. So we have stopped that.
True, I come from industry, I'm not from academical research institutes. So in the industries, if you see this kind of product, mainly from the globally, you go to Europe or any country, even the Middle East countries, there's tremendous demand of such works. And those countries are primarily looking or greatly looking to the countries like us for this kind of work and this kind of product. So I believe if you have some data on comparison with the classical market, what are available in the market, and what sir said about the stability, this can go great for commercialization in coming days.
Yes,
Some
sir.
kind of effect.
Yes, sir. Because we have to do, because if we have to commercialize, we have to do, and we have to justify that also. So if we have to go for the product launching, if any of
We
the
have to do
company,
that.
then we have to test that we have been
So regulatory authorities, they do not expect the product to work as good as classical antimicrobials. Even 60-70% also the regulatory authorities are saying that please come out with such product.
Yes.
So I congratulate you for such a product, and I hope it goes further and it goes for commercialization.
Yes, sir.
Thank you, Dr.
Thank
Samara.
you.
Thank you so much. Nani, what is your message from your research to the public?
Go nano, go green. Every time what happens, we are restricted to the we are restricted to only that much. I have said to, okay, the biochemists, they are only restricted to the laboratory only. But when it goes to the lab, that is our purpose. So we should go take our laboratory findings to the lab. So that one step I have forwarded in that case.
Thank you for your presentation.
Thank you, sir. Thank you.
Offering for presentation of master's thesis award.
Madam, thank you.
Thank you,
Nice
sir.
presentation.
Seasonal variation on oxidative stress biomarker and removal gene expression in relation to seasonal quality. Livestock play a major role in the Indian economy. Among livestock, goats have tremendous potential to be projected as a future animal for rural prosperity under the changing agroclimatic condition and depleted resources. According to BHS 2019, India ranks second in the goat population with 148.8 million goats. Goats undergo several types of stress, but the oxidative stress is the most common. Oxidative stress is caused by the elevation of the reactive oxygen species or by the depletion of the antioxidant defense mechanism. Removal gene is one of the most important proteins to modulate the reactive oxygen species. The objective of my thesis includes to study the seasonal changes on seminal antioxidant biomarker status and seminal gene expression in buck sperm. That location of my work was Department of Veterinary Physiology and Biochemistry, College of Veterinary Science and Animal Husbandry, NTVSU Jabalpur, and the duration of work was 6 months. Next. 12 adult breeding bucks of Barbary and Sirohi breed were used, and all the breeding bucks were maintained at the Amanala Goat Farm, NTVSU Jabalpur. The age of the animal was in between 1.5 to 4 years. This picture represents the flocks of the Barbary and Sirohi breeds at Amanala Goat Farm during the sample collection. Four ejaculate fortnightly collected during the summer season and winter season of both the breed Barbary and Sirohi. So the total number of semen sample includes 96 samples. 12 semen samples was collected by using the artificial vagina method, and after immediately the sample collection ejaculates was transferred to the laboratory for further evaluation. Parameters of study include the climatic condition during the study period, including average daily ambient temperature, relative humidity, sunshine hours, and temperature humidity index. Reproductive parameters include the scrotal circumference, which is measured by the measuring tape, and the scrotal temperature by thermoscanner, and then evaluation of semen by as the described method by the Humphreys and Humphreys 2000 includes semen volume, semen pH, individual motility, mass motility, spermatozoa concentration, live and dead count. Estimation of biochemical parameters, enzyme activity and oxidative stress parameter in seminal plasma. Biochemical parameters include the total protein concentration by Laurie et al. 1951 method and albumin concentration by Thomas et al. 1972. Total cholesterol and calcium by the Aram Manning diagnostic kit method. Next. Seminal plasma enzyme activity includes the alkaline phosphatase, aspartate transaminase, alanine transaminase, and lactate dehydrogenase by the Manning kit method. Next. Oxidative stress marker parameter includes superoxide dismutase activity by the Madesh and Bal Subramanian 1997, by the slight modification and catalase activity by the Both 1991, reduced glutathione concentration by the Prince and Luz 1969, and lipid peroxidation by Suleiman et al. 1996, and total antioxidant capacity by Nazi et al. 2020. For the expression of the Romo1 gene, first total RNA isolation by the triazole method, and after that the cDNA synthesis by kit method, and after that the quantification of the Romo1 gene by taking our GAPDH as a housekeeping gene which amplicon size is 168 base pair, and Romo1 gene the amplicon size is 153 base pair. Statistical analysis was done with the help of the SPS software using repetitive measure ANOVA statistics. The result of my first objective. Next slide. Average meteorological parameter. The average meteorological parameter was recorded the Meteorological Center Anandnagar Adharta, which includes the temperature maximum, temperature minimum, dry bulb temperature, wet bulb temperature, mean relative humidity, and temperature humidity index. And we found that a higher PHI was recorded during the summer season as compared to the winter season. Next. The reproductive parameter of Barbary and Sirohi breed during the winter season and summer season, there is no significant difference in between the breed, but there is a significant difference in between the seasons. The summer season has higher scrotal temperature as compared to the winter season in both the breed, and the scrotal circumference is higher in winter season as compared to the summer season in both the breeds. Next. The graph represents the same. Next. Semen physiological parameter of Barbary Sirohi breed during summer and winter season, which includes semen volume, pH, sperm concentration, mass motility, individual motility, and live sperm count. So, pH and mass motility is non-significant in between the breed and in between the season, but the semen volume, sperm concentration, individual motility, and live sperm count was higher in winter season as compared to summer season in both the breeds. This is the color of the semen, a collection of the semen which is creamish white. The plate represents the sperm concentration in Newbar's chamber, and this is the live and dead sperm by eosinophilic stain. The graph represents the same. Table 5 represents the seminal plasma biochemical parameters of Barbary and Sirohi breed during the summer and winter season, which include total protein, albumin concentration, total cholesterol, and calcium. There are no significant differences between the breed for 4 of the parameter, but there are significant differences between the seasons. The winter season has more total protein, albumin concentration, total cholesterol, and calcium as compared to the summer season. The graph represents the same. Seminal plasma enzyme activity estimation of Barbary and Sirohi breed during summer and winter season include ALT, AST, ALP, and LDH. There is no significant difference in between the breed and in between the season for the ALT, but there is a higher the value for the AST in winter season as compared to the summer season in both the breeds. But the ALP or LDH was higher in summer as compared to the winter in both the breeds and in both the breeds during the summer season. Graph represents the same. Several plasma oxidative stress parameter of Barbary and Sirohi breed during summer and winter season include SOD, catalase, reduced glutathione, MDA, and THA. The SOD, superoxide dismutase, and MDA was higher in summer as compared to the winter in both the breeds, so there is no significant difference between the breeds. But the catalase activity and reduced glutathione and total antioxidant capacities were higher in winter as compared to the summer in both the breeds. Graph represents the same. Objective 2, to study the seasonal influence of Romo1 gene expression in buck sperm. There is a mean value of relative expression of Romo1 gene in Barbary and Sirohi breed during the summer season and winter season, and there is And there is 0.89-fold reduction of bromobenzen expression in winter season in both the breed and 0.84 reduction in Sirohi breeds. There are the PCR amplicon of the GAPDH loaded on 2% across gel and amplicon for bromobenzen loaded on 2% across gel, in which the size of the GAPDH was 168 base pair and the size of the bromoben was the 153 base pair. Primary efficiency plot for RT-PCR, which is the R value is 0.99. Amplification plot of the samples. Next. Melting curve. Now I am concluded my thesis as climatic condition affect the semen quality. High quality is produced during the winter season as compared to the summer season, and high THI during summer affect the different seminal biochemical parameters, concentration and seminal plasma enzyme activity. Summer also impair the antioxidant defense mechanism of the seminal plasma, and seasonality affect the Romavan gene expression with down regulation in winter and up regulation in summer. Suggestion: there is need to extend the present work on large number of animals in different seasons to optimize the semen quality and fertility. There is need to assessment of the Romavan modulation to improve the semen quality and fertility in goat buck. There is need to study the daylight and light intensity effect on oxidative stress biomarkers and Romavan gene expression in goat buck semen. And there are two publications of my work. One in reproduction in domestic animals, which is mass rating is 7.4, and next the semen quality correlation with seminal biochemistry and enzymatic profile in the Journal of the Indian Journal of Animal Research. Thank you.
Thank you. Any point to be discussed or asked?
You selected Romo gene. What is the function?
Sir, Romavan gene is a modulator of the reactive oxygen species. When means there is a relation of the oxidative stress is increased when the expression of the Romavan gene is increased. It is a mitochondrial membrane protein which accelerates or modulates the formation of the reactive oxygen species. So we can connect them to the oxidative stress. Sir, Romavan gene is a protein. Gene selective. Reactive oxygen species modulator 1. No, sir, AST is higher in winter season. Sir, this is a contract. So, my question is that. Ma'am, actually during the summer season, the management of the goats in goat farm is not very much. Ma'am, due to the effect of the environmental stress and high PHI, the animal goes in the stress. So these all parameters are increased, and like enzymes, these are intracellular enzymes. When there is leakage in the membrane, they are come outside. So the elevation is high in summer as compared to winter.
Next presentation by Vivek Kumar Pandit for MBSc, base MBSc thesis.
Very good afternoon, respected chairpersons, reporters, delegates. I am Dr. Vineet Kumar Pandey, currently a student, PhD scholar at Indian Veterinary Research Institute. Today I am going to present my best thesis award seminar titled Comparative Evaluation of Circulating Perilipines 1 and 2 in Normal and Canine Mammary Tumors. So canine mammary tumor is a common neoplasm in the female dogs, and it accounts mainly for mostly for the 40% of the total cancers found in the dogs. So one of the most important hallmarks for this cancers is the altered lipid metabolism. So this lipids it helps in the signaling of the various molecules. So the adipocytes and the pre-adipocytes are the within the breast tumors environment plays an important role in the aggressiveness and the metastasis. So the lipid droplets accumulates in the variety of the cancer cells, which includes the breast cancer, lung cancer, etc., which suggests the role of the lipid droplets in the cancer metabolism. So lipid droplets are the small intracellular organelles which consist of the neutral lipids such as the triacylglycerols, diacylglycerols, and they are bound with the phospholipid monolayer, and it consists of several proteins, and one of the proteins is called as the perilipin proteins. So next. So perilipins are also called as the PAD family, and it consists of 5 proteins, that is perilipin 1, 2, 3, 4 and 5, and it plays an important role in the lipid biogenesis, stabilization and lipid trafficking. This lipid adipocytes in the mammary tissues play a significant role in the lipid metabolism, which plays as emerging important player in the canine mammary tumor. So this canine mammary tumor resembles the human breast cancer in histology, clinicopathology, molecular features, etc. So, despite these similarities, species-specific differences highlight needs for deeper understanding of the CMT biology to understand the role of the lipid droplets and associated proteins in the canine mammary tumor. So, we found a knowledge in gap that the intracellular role of the planes is tissue specific and it is not found in the circulation. So for this we hypothesize that there is differential circulatory perilipins in the CMT in comparison to normal control serum samples. So our aim of this study was to evaluate the levels of the circulatory perilipin 1 and 2 in normal and canine mammary tumors. Next. So for this we have 2 objectives. That is, first is the expression and purification of the recombinant canine perilipins 1 and 2, and second is the detection of circulating serum perilipin 1 and 2 in normal and canine mammary tumor. So technical program for the first objective. So first we designed the primers for perilipin 1 and 2 with the primer 3 software, and we amplified this gene with the DNA of the canine mammary tissue. Next. Second, we cloned these canine perilipin 1 and 2 in the pet vector and the sumo vector. So we target the DNA target was bulk amplified and purified and RE digested. Then the DNA fragments were purified with and was ligated into the RE digested PET32A and sumo vector. The recombinant vector was then transformed into the BL21 cells and the E. cloni cells, and was then grown into the LB broth in ampicillin and kanamycin antibiotics. Then the colonies were picked and confirmed by the colony PCR, and further it was RE digested. Lastly, the confirmed colonies were cultured in the LB broth, and thereafter small scale induction was done with ITPG and granules. For the second objective, we did the dot blot and the western blot analysis. So serum samples were collected from the PVCC IVRI, and first the protein concentrations was measured with the BCA method. These serum samples were screened by the dot blot analysis. Then further, the western blot analysis was performed for the screened dot blot samples. Then the western blot was probed with the anti-plin-1 and anti-plin-2 antibodies. Next. So for the objective one, next. So these are the primers for the canine perlipin 1 and 2. Next. So first we have amplified the canine this perlipin 2, which was having the 794 base pairs. Then next we bulk amplified and digested with the ECOR1 and HIN3 restriction enzyme, followed by the same digestion with the PET32A vector with the same enzymes. Then we ligated the PLIN2 gene into the PET32A vector. Next. Then this recombinant plasmid was then transformed into the E. coli cells, and then it was confirmed with the colony PCR. This after the colony PCR confirmation, we then digested the recombinant plasmid with the same enzyme, that is the ECOR1 and the HIN3, and we found that it was our product, that is the PLIN2, was ligated successfully in the PET32A vector. Next. So after this confirmation, we went for the transformation of the recombinant plasmid into the BL21 cells, which is the expression host, and we then induced our culture with the IPTG, and we found the recombinant protein at the 46 kilodalton size. Next. So after the confirmation of the proteins, we further purified our recombinant protein with the 8 molar urea, but however we could not get successful. So we further eluted it with a 6 molar guanidine hydrochloride buffer, and we found that the recombinant protein is present in the guanidine hydrochloride lysate. Next. So further we went for the protein extraction of the proteins from the gel with the native gel, and then we have sent for the antibody preparation. Next. So this is the ligation of the perlipin 1 in the sumo vector. So first we amplified the gene with the PLIN1 gene, and it was then digested with the BGL2 and NOT1 restriction enzymes, and this was then ligated into the PET sumo vector. And next, please. So then the recombinant plasmid was then transformed into the E-clonized cells, and then the colonies were picked and were tested for the colony PCR, and we successfully verified the recombinant plasmid by digesting with the same enzymes. We separated the gene. Next, please. So further we induced this E-clonized cells with the L-rhamnose, and we found that the recombinant protein was induced at 29 kilodalton. Next, please. So now comes the second objective, that is detection of the circulating serum perlipin 1 and 2 in normal and canine mammary tumors. So we have took 30 dog serum samples from CMT and 12 from the normal, and these are the corresponding protein levels in the serum sample. Next. So first we screened for the perlipin 2 by the dot blot analysis, and we found that the PLIN2 is found in almost all the CMT samples and in some of the normal serum samples. Next, please. So further we went for the Western blot, and first image is seen as that we have done is the Ponzo staining, and then we have successfully done with the anti-PLIN2 antibody, which is commercially available, not the in-house grown. Next, please. So this is for the PLIN1. We have screened with the dot blot analysis for the PLIN1, and we didn't got any screening for except for the 1 or 2 in the CMT samples. Next please. So this is the Ponzu staining for the perlipin 1 and the corresponding figure shows the plin 1 antibody. So summary, so we have generated the recombinant plasmid of the canine plin 1 and plin 2 gene by cloning into the ramnose, p-ramnose sumo vector and the PET32A vector. The nucleotide sequencing was confirmed by the presence of canine plin 1 and plin 2. The recombinant canine perlipin 2 protein was generated as insoluble form and the protein was solubilized using the 6 molar guanidine hydrochloride buffer with 3% SDS and purified with the gel extraction after reversible zinc imidazole staining. The immunoblotting analysis confirmed the antigen identity of the recombinant canine plin 2 proteins. So dot plot analysis employed the initial screening of the circulatory perlipin 1 and 2 in the serum samples of the normal and canine mammary tumor. So for this we had took the 30 CMT samples and 12 normal samples, and the plot developed with the commercially available anti-plin 2 showed reactivity in all the 30 CMT samples and in 3 to 4 normal samples. The reactivity in the control may be possibly due to many factors which might increase the plin2 expression, such as the lipid disorders, sex, age, acute or systemic inflammation, liver or kidney disease. And control were apparently healthy and no report on health status was reported. Next please. So further, the circulatory perlipin 1 and 2 were characterized by the Western blot analysis using the commercially available antibody plin 1 and plin 2. The protein loading controls were checked by the expression of the transferrin using the anti-transferrin polyclonal antibody. The plin 2 band were detected in almost all the serum samples, while the circulatory plin 1 protein bands could not be detected in the present plin 1 antibody. Next please. So I will conclude. So this is the study we have done for the first time, that in the serum the perlipin 1 and 2 in the normal and canine mammary tumor. So recombinant canine plin 2 protein generated for the raising hyperimmune sera, and circulatory form of the canine perlipin 2 was detected in the CMT sera. So our findings are in line with the previous reports on the secretory form of the plin 2 detected in human patients with renal and colorectal carcinoma and the female patients with lipid disorders and multiple organ failure. Thank you.
Thank you, Pandey, for your presentation. Now, his presentation is open for discussion. Any point to discuss?
A nice presentation, and I want to know what are the future perspectives for this study.
And this is basically the study for screening of the canine mammary tumor with the serum samples. So we could not generate our in-house antibody, so we have took commercially available antibodies. We are in process of making the recombinant protein, producing the recombinant antibody for our work.
Pandey, can you state some basic properties of perlipin?
Sir, it helps in the lipid biogenesis, and it helps in the stability of the lipid droplets. So basically, the lipids when they are stored in this lipid droplets, like basically this triacylglycerols, so they are stored in that.
What is the molecular weight?
What's it?
Molecular weight of that protein, perlipin?
Sir, molecular weight for perlipin 1 is 52 kilodalton, and for plin 2 it's 48 kilodalton.
Number of amino acids present in perlipin?
So 300 amino acids.
Okay, thank you.
Thank you all for your kind questions. And this is perhaps one of the best presentation award. We used to know how the future generation is progressing, how the future scientists are working. So this is the time to end this session. Our teachers of all teachers, Professor Amalandu sir, to come up with the dais to felicitate chairperson. Please switch on the lights. So it's a great prestigious moment to have Amalandu sir with us. He is our teacher and teacher of all the person. So I'd like to felicitate Chairperson Amalandu sir. Thank you, sir. I'd like to greet another chairperson, Anita ma'am. Anita ma'am, please come. Thank you, ma'am. Please, sir. I request sir to felicitate our rapporteurs also. Please come to sir. Please come.
Tell her name.
Kavita Rawat. Please felicitate Kavita Rawat, Assistant Professor in MOM, Department of Biochemistry. Thank you. Thank you, sir.
Respected Vice Chancellor, all the PGs, it is a great opportunity for me to hand over the awards to the different chairman, co-chairman, rapporteurs, and others. So I thank the organization to give me the opportunity to stand by you to felicitate you. Thank you.
This marks the end of this session. This is the last presentation. Thank you, sir. Thank you all.
What is next?
So this is the last one.
Next is what?
Next, I think we should move for the lunch. Please, Sam.
Then tell everybody.
Yeah. Please move to the lunch. This session has ended. Let's welcome to the lunch session.
আমরা তো মালদা থেকে